Our next slide shows physically what it looks like. And so you can see the various components that have been isolated and have had X-ray crystallography done on it. This A subunit right here of the chloroplast version still does not have its X-ray crystallography done. This is the same protein that hasn't had its structure done in mitochondria either. So let's take a look at this. So here, in terms of orientation, the lumen, this is inside the thylakoid sacs. This is the liquid in here. The thylakoid membrane is this bilayer that most of these proteins are embedded in. And out here is the stroma, the negative side with regard to the proton gradient. And it's out here in the stroma, that's the liquid that bathes the outside of the thylakoid sacs. It's out here that the enzymes of the Calvin cycle are, that do the synthesis of CO2 and water to form sugars. So, plastocyanin, this is a small protein. You'd expect a protein because a protein that is tiny, 10,800 is a tiny protein, can diffuse fast. You don't want some big thing slowly moving from one part to another. It also has a copper ion, that's the cyanin part, so there's a copper that goes from +2 to +1 if it picks up an electron. So the protein part is holding the copper and allowing that electron to bind, and the protein part is what recognizes these two binding sites, one on photosystem 1 and one over here on the cytochrome B6F complex. So the protein is what docks specifically at these two complexes. If this was just a copper ion, it would just diffuse and never know where to bind correctly. So this is one of the mobile carriers. It's not carrying a proton. It only carries one electron. And so it's going back and forth here between PS1 and the cytochrome B6F complex. The cytochrome B6F complex, again, is where you bring your hands together and form two caverns. That's where the Q cycle is occurring. Ferredoxin is about the same molecular weight as this. These are rounded here. That's this tiny protein, and it's out here in the stroma, and it has an iron-sulfur complex that can only carry one electron. And so what it does is it carries the electron back and forth here. It's taking the electron to this enzyme here, which is the enzyme that reduces NADP plus to NADPH. So that's this enzyme, this flavin NADP oxidoreductase right here. Ferredoxin can only carry one electron at a time. So it's carrying the electron from PS1 over to this enzyme. NADPH requires how many electrons to be fully reduced? 2. So you're going to need 2 different ferredoxin molecules, docking, etc. So the protein part, the fact that it's small, means it can diffuse quite rapidly in this liquid area. But the protein part is also what is docking very specifically to this enzyme out here and to PS1, photosystem 1. So that's our second carrier. Again, it does not carry protons. It just carries one electron. And the last one is plastoquinone B, PQB. And this is the one that looks very similar to coenzyme Q in the mitochondria. it's mostly hydrophobic. It has a little polar part that carries the electron. And so plastoquinone B is going back and forth here between photosystem 2 and the cytochrome B6F complex. So plastoquinone B, because it's mostly hydrophobic, is imprisoned in this thylakoid membrane. It's never getting out. But it can carry either one electron or two, and it always carries that electron with a proton. So it's carrying it as H dot or two H dots. And I'll show you the structure here in a minute for plastoquinone B. But the point is it's very similar to coenzyme Q in the mitochondria. Photosystem 2 has, as part of its structure here, this oxygen evolving complex, OEC. We'll talk a little bit more about that in a minute. And this is where water molecules somehow come in and have electrons and protons stripped off of them. And so the protons are helping to create a proton gradient. So in the chloroplast, inside the lumen part, there are two sources of protons being created. One is from the Q cycle here, because every time this thing binds, an electron goes up to the electron carrier, the other electron goes into the other cavern, and you've got the two things, the risky iron sulfur proteins going up and down like this, and they're dumping protons. The Q cycle is dumping protons from plastoquinone B into this chamber, this lumen. The protons are also coming over here from water being stripped. So you've got two sources of protons, and those protons are going to go over here to the ATP synthase, which looks pretty much the same as the one in the mitochondria. So protons are going to go up a half channel here, around the merry-go-round, and then out. And in that process, they drive the wheel, which drives the crooked gamma subunit, 120 degrees at a time, 3 clicks. And so ATPs are being made out here in the stroma, and this enzyme is making NADPH out in the stroma, and that's exactly where the Calvin cycle enzymes are that need both of those in that 3-to-2 ratio. So you see how structurally everything is where it should be. Okay. So let's look a little closer here at photosystem 2, PS2. So in photosystem 2, we've got these two chlorophylls. The dots here represent, in Leninger's photograph, a magnesium. So these chlorophylls are what I was talking about last time. My hand is the porphyrin ring of one chlorophyll. My other hand is the other porphyrin ring. And these two chlorophylls are held by proteins so close together that they act as one. And so energy is being fed to them to pop an electron. So the numbers here represent the sequence of events in less than a millisecond. So step number 1 is that an electron is being passed, the blue wave is an electron, being passed to pheophytin. Pheophytin here does not have a magnesium in it, that's why there's no dot there, but it's being held by the protein. The electron is going through the backbone and R groups of the protein, but when it goes through pheophytin, we can say, oh, I can see this because the spectrum shifts. And now that electron goes through plastoquinone A, that's the one that is bound by a protein. It doesn't move, but it gives its electron over here to plastoquinone B. This iron represents an iron sulfur center that's in this protein. Plastoquinone B will then diffuse away. So the first plastoquinone is fixed, it doesn't move. The second one is what picks up an electron and a proton, H dot, and then it can pick up two, max of two, and that's going to float away in the membrane. It's hydrophobic. So I kind of, this is 1, 2, and then 3 is actually up here, but I just wanted to finish down here what's happening to that electron. It's winding up on plastoquinone B. Meanwhile, up here, waters are the ultimate electron donor. And this structure here has taken many years to figure out that there's actually all these atoms that are there. Initially, it was thought that there was only 2 manganese, and then over the years, more and more atoms were discovered as the X-rays, the crystals were better, and so you could see the position. And so you actually have this complicated structure here that I'll show you a little bit more in a second, that has 4 manganese, a calcium, and some oxygen atoms in it. Not oxygen molecules, but oxygen atoms. So it kind of looks in this diagram here to me like an upside down chair. And somehow this group of atoms, along with the proteins that are holding it, are able to take electrons off the water, and when those electrons are stripped off, it releases protons. So this is showing you the release of protons. This would be called an enzyme active site. Protons are being stripped off as electrons are. And this tyrosine is part of this protein here. It's a residue that is part of the circuit by which the electrons are fed back one at a time to P680. Remember, when P680 receives energy from the environment, from supporting molecules that are grabbing photons of light, those excitons are being sent to this binuclear, I can't just call it a binuclear site, to this dimer, chlorophyll dimer. As it pops an electron, then this thing becomes, remember, P680 plus. Give me that electron. So this is the thing that is really thermodynamically sucking electrons away from water. This thing has such a thirst. When it pops that electron, it becomes a nasty P680 plus, and it's going to get its electron. So one at a time, somehow these electrons are stripped off of this complex here, one at a time. When an electron comes here, boom, it can pop another electron. Okay, so here is a close-up view of this chair assembly. It's turned around compared to the previous slide, 180 degrees. These are residues that are there by the protein, but you can see that this thing, this chair here, has 5 oxygens, a calcium, and 4 manganese. But we are still, in my opinion, a long ways from still understanding exactly how it works. The reason being several. One is that there are waters everywhere, and they move so fast. They're parts of protein structures. You know, if my fist is a protein, there are shells of water bound around it all the time. And so there's so many waters, it's hard to know, hey, which one of you is actually having your electrons and protons stripped off. So that's one problem. Another problem is electrons move incredibly fast, and protons hop. And so the whole thing is very fast and very difficult to detect. I'm sure we will with better technology years from now, but right now it's still a mystery. So there's a lot of research being done on this, but hasn't been solved. So this is just a model of how this thing is thought to occur. Again, this is the chlorophyll dimer up here. Somehow this complex, the 0 means it's in its ground state, And as protons come off, again, this is a model, we don't know how they come off. As protons come off, this thing is capable to going up to a +4 state. So that's one of the cool things about metals. They can take on various oxidation states. And so the entire complex here is being raised to a much more positive value. But ultimately, it's this guy here, this dimer, becoming P680 plus, that is just ripping the electrons off of this thing. This thing is like a black hole here. So I wrote this, but it's on your notes. Again, it just says that we're not quite sure how this OEC works. Okay, so this is the structure. I would only ask that you recognize it. But this is plastoquinone. And so plastoquinone A and plastoquinone B have this structure. The only difference is one is bound by a protein and the other is free to move in the membrane. So once again, we see that there's this slightly polar part right here that can grab an electron, the quinone part, the aromatic ring with the double bonded oxygens. When one electron comes on, it's momentarily stabilized because the electron is spread out over many positions on the molecule. Otherwise, it'd be a free radical that would be hard to deal with by the cell. And this hydrophobic tail is very similar to the hydrophobic tail on coenzyme Q. So there's 9 of these isoprene units. So this thing can go stepwise and pick up one H dot, become a semiquinone. I'm just going to show the fully reduced form. So just like coenzyme Q can exist in 3 oxidation states, up here is the fully oxidized one, down here is the fully reduced. But notice that neither one of these has a charge. That's why it can operate in a membrane. So coenzyme Q from the mitochondria looks very similar, and the modifications I'll just show you in green, they're fairly minor. There's an oxygen here between this carbon and this methyl group, another one here, and finally a methyl group, and that's it. So it's pretty much the same molecule. It's just been adapted to a different environment. So somehow this works a little bit better than coenzyme Q in the chloroplast. So if you want something to carry electrons and protons fast, this is pretty elegant because this is just a small organic molecule. It's not a protein, which is much larger. It can diffuse quite rapidly in the thylakoid membrane. Now going back to the Z scheme, let's look at this cytochrome B6F complex. I've already told you it looks very similar to the complex 3 in mitochondria. It's got the 2 caverns and the 2 risky iron sulfur proteins going up and down. And so they're just showing you the cavern here. Plastocyanin is not part of this complex. They're just showing plastocyanin here is analogous to cytochrome C in the electron transport chain. Plastocyanin is simply docking, picking up the electron that this funny protein, again, this is the risky iron sulfur part right here, all part of this weird protein that has that long flexible neck that wraps around. It's just going to lift that electron from this binding site up to the next protein that sends it up to plastocyanin, and plastocyanin, presumably when it picks up that electron, it changes its conformation, releases from the cytochrome B6F complex, and then can go dock where it's supposed to dock. So I just want to point out here that there are some carotenoid pigments in here. Remember we said that those are pigments that can absorb light in a leaf, and cells use what they have, but there are a couple of carotene molecules that are in here, presumably acting as part of the electrical circuit. So that's a slight difference from the mitochondria. Mitochondria don't have carotene molecules. But the overall gist is that it's the same. And so here is just a abbreviated form. Here is the Q cycle, and the reduced form comes in. One electron goes up to plastocyanin. There's a copper ion in the protein. That's how it gets its name, cyanin. It's the copper here that carries that one electron. It'll then diffuse away. The other electron goes over into the other chamber, other cavern. Okay? All right. So now let's go to our last set of slides, actually, but we're not going to finish it today. This is fine. So we need to talk about briefly photosystem 1, and then get into the carbon assimilation reactions, better known as the Calvin cycle. So here is photosystem 1. All of this, all of these proteins, etc., that are part of this photosystem 1. Here it is, embedded in the membrane, the thylakoid membrane. So here comes plastocyanin with its copper ion, carrying one electron. Plastocyanin is not part of photosystem 1. This is the protein that picked up the electron from the cytochrome B6F complex, and now it's docking in a particular region. So this is the one electron carrier. It is supplying the electron that's disappearing because this photosystem has its own chlorophyll dimer here. So again, the reaction center, two chlorophylls acting as one. When the energy from all of these accessory pigments, when that energy is funneled to me, the reaction center, I pop an electron, but I'm dependent on plastocyanin to give me a new electron so I can pop the next one to the higher energy level. Is that okay? So that's why this is all part of a circuit. An electron gets popped to a higher energy level, the chlorophyll dimer says, I need another electron. So that electron has, it turns out, one of two paths that it can go to, but it winds up going through some iron-sulfur proteins that are part of Fa, Fb, these are iron-sulfur proteins. It winds up going to ferredoxin. Ferredoxin is one of those other circled blue proteins on that Z scheme. Ferredoxin is not part of photosystem 1. So it's docked here. As a protein, it has weak bonds that know where to dock very specifically. And so this thing picks up one electron, no protons, and it stores it on its iron-sulfur center. And so this thing, once it picks up its electron, presumably it changes its conformation, releasing those weak bonds. And now this goes over to the enzyme that's going to synthesize NADPH from oxidized NADP+. So two different ferredoxin molecules have to go and dock with this enzyme to make one molecule of NADPH. Okay? So this is a summary here of what's going on. First of all, let's get rid of the problem that always seems to exist with Leninger, and that is The protons are not going straight up the middle of this ATP synthase. They go halfway around and then out the other half channel. Also, ATPs are not being made out here. They're made more in here at each of the 3 dimer active sites, okay? So the model is the same as what we correctly talked about with the mitochondrial ATP synthase. So CFO and CF1, the C just stands for the chloroplast. And so the subunits are a little bit different, but the overall architecture is the same. So what I wanted to show you is that you can see that the cytochrome B6F complex is donating some protons, and this manganese is representing that chair assembly, the oxygen evolving complex, and together they're providing the protons that are running the synthesis of ATP through this structure, okay? But you've got 4 protons coming off of 1 PQH2. Remember, this is only possible because of the Q cycle. So we talked about that in the mitochondria. In the Q cycle, 2, in this case, PQH2s are consumed, but 1 is made. And every time a PQH2 dumps off 2 protons, well, you got 2 of them, you're getting 4 protons and not just 2. So in this diagram here, the way they're getting 4 protons when this thing only has 2 is it's running the Q cycle, the very same Q cycle. So go back to your notes that we discussed for the mitochondria, okay? That'll make it clear as to why life does it that way, but I'm trying to explain why Leninger puts 4 protons here. 1 PQH2 winds up giving 4 protons because of the Q cycle. Is that okay? And here, plastocyanin, there's going to have to be 2 of them to provide enough electrons to create NADPH. But once again, one of the nice things about the diagram is it shows us the orientation of this thylakoid, and the NADPH is being produced out here in the stroma, and so is the ATP, because that's where the Calvin enzyme cycle enzymes are that need this to generate the carbohydrate. Okay, so we're not memorizing this equation, but I want you to understand it, okay? Again, what this says is that everything is linked together to create 3 ATP and 2 NADPHs. So the operation of photosystem 2 and 1, along with the cytochrome B6F complex, together they're making 3 ATP and 2 NADPHs. So this is where you see the photosystems and the apparatus of the Z-scheme making the 3:2 ratio of these 2 components. These are both high energy molecules. Cool? Because that's exactly what's needed by the Calvin enzymes out here. Okay? They work completely together. The enzymes out here, they're going to take all that ATP and NADPH, are just recycling it. Okay, I think we're going to stop there today so you can get out of here and enjoy the weekend. And we'll meet again on